Life will find a way through night and day, living in true awareness, come what may. We are the master of our fate, by willing, praying, standing right. We learn, we try, we aim higher, with true courage a greater gain. By obeying we grow, by trying we grow, to reach the help from heaven above. Miracles come through faith and hope, to those who dare, to those who fight. Miracles happen to those who believe. Step by step we fight with bravery, hearts wide open, never alone. In silence or in battles cry, the answer comes when spirits fly. We strive, we thrive, we heal the soul, and let the highest power unfold. By obeying we grow, by trying we glow, to reach the help from skies above. Miracles come through faith and hope, to those who dare, to those who try. Miracles happen to those who believe. Not by chance, but by design, a path is drawn, a sacred sign. Hold the line, don't let it fade. History in the making. By obeying we grow, by trying we glow, to reach the gift from skies above. Miracles come through faith and hope, to those who dare, to those who fight. Miracles happen to those who believe.
Would like give introduction, Professor? It's okay?
Yes, I'm fine, thank you.
Okay. Okay. Hello. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon and evening, Indonesia. Good morning, England and United Kingdom. Good very morning, USA. Good day for our friend from all the rest of the world. It must be evening and Friday night in Pacific, Australia and New Zealand, noon, afternoon and evening in Asia, morning in Europe and Africa, and morning or early morning here in Boston, and also in North America, Central America, and South America. First and foremost, thank you everyone for coming. Welcome to this international webinar that is attended by participants from many countries of many continents around the globe. We are gathering this day, all nations, citizens of the world, spreading care, love, and peace to be united for helping the people against blindness. Welcome to our 36th episode of Distinguished Maestro Lecture. I'm Gede Pardianto from Indonesia. Let's start the webinar. To honor the origin country of this webinar, please pay attention to Indonesian National Anthem. Thank you so much. To honor the origin country of our distinguished speaker, please pay attention to British national anthem, God Save the King. Thank you so much, ladies and gentlemen. The super maestro this time will be Professor Harminder Singh Dua, a legend, a friend of mine, multiple awards winner, champion, the man, and the grandmaster. He is professor of ophthalmology at the Queen's Medical Center, University of Nottingham, consultant ophthalmologist, Nottingham University Hospitals, honorary professor at Universitas de la Suisse, Lugano, Switzerland, and honorary professor at the Research Institute of Ophthalmology, Cairo, Egypt. He was president of Royal College of Ophthalmologists, the president of Cornea, and European Association for Vision and Eye Research. He was made Commander of the Most Excellent Order of the British Empire by Her Majesty Queen Elizabeth II in her 2019 birthday honor list. He served a full term as the High Sheriff of Nottinghamshire. He was also awarded the National Health Service Parliamentary Lifetime Achievement Award for the Midlands. He was a speaker and moderator in more than 100 scientific meetings around the world. He authored hundreds of journal articles, editor for multiple journals, and editor-in-chief for Eye, the most legendary journal of ophthalmology. Editor and co-editor for dozens of books, author of multiple books and book chapters, one of the most respected and high-impact author with more than 35,000 of citations around the world. He is awardee of more than 150 awards. His discovery of the Pre-Descemet layer or Duas layer has had significant impact on lamellar corneal surgery and corneal pathology. Wow, that is exceptional and so awesome. At this very moment, he will present a very interesting topic with the title, The Discovery and Impact of the Pre-Descemet Layers or Duas Layer. It will be a really remarkable topic. Now, we are learning from the man who is truly expert in this field. So be prepared. Fellow ophthalmologists, let's pay attention to Professor Harminder Dua. Harminder, we are counting down for you. 5,
Thank you.
4, 3, 2, 1. Time is yours. Lift up, applause. Godspeed, Professor Harminder Dua.
Well, good morning from the United Kingdom, and thank you very much. I will share my slides with you and get that let's get this going. So, yes. And this, while I tidy up my screen, get everything in order. Thank you very much for that very kind, very generous introduction, and for the honor of giving me this opportunity to present your Maestro lecture and be part of your illustrious list of speakers that you presented in the introduction. So my topic, as you have mentioned, is a little account on how this layer was discovered and the impact it has had on corneal surgery and corneal pathology. More than 100 years ago, corneal transplants were started. We had xenografts first and then human corneal transplants. And some of the very first corneal transplants were square grafts, as you see over here. And when I, long time back, was preparing my talk and my son was asking, what's this? I told him, this is what happens to your eyes if you watch too much television. But that is how this started. And despite the time interval between then and now, many significant problems remained. For example, rejection and non-rejection graft failure is a problem. When there is stem cell deficiency, the grafts fail. Even when the graft is nice and clear, we have graft astigmatism can be very high, and the outcomes may not be good until more surgery is done or other devices are adopted. And no matter what we do with a penetrating graft, the graft-host junction remains forever weak. Even with a little trauma, decades after the graft, it can rupture with disastrous consequences. And this cartoon illustrates, as a method for checking cornea grafts, this is crude but is effective. So no cornea graft can survive a trauma like that. So the quest was always there to try and change this. And the answer came not so much from scientific advancements in immunology and immunosuppression, but from surgery. And the basic question was that if the Descemet's membrane and stroma are not involved, then why change them? Like in keratoconus and stromal dystrophies and scars, and here you see a patient with granular dystrophy, we can take the stroma and the epithelium away, leaving the patient's Descemet's membrane endothelium behind, and that way we forever remove the risk of graft failure due to endothelial rejection. And the converse was also true, that if the endothelium is affected, then why change the rest of the stroma? We can remove the affected endothelium Descemet and replace it, for example, with the operation of Descemet stripping endothelial keratoplasty or with DMEC, and then you have no sutures, you have no graft-host junctions, so those problems of suture-related complications, astigmatism, etc., are all eliminated. So my story begins with the operation of DAG. We all know that there we make a deep trephination, and then we have different ways by which we can separate the Descemet's membrane from the stroma. And this is the most popular method, the Unwar's big bubble technique. A drop of helon is placed on the corneal surface, a needle or a cannula is injected in the stroma, air is injected, deep trephination has been made, and you see everybody knows what happens. You will find that the air will spread as a white ring. It will go up to a certain limit. It very rarely goes up to the limbus, 8 or 9 millimeters. You take the top stroma off, you puncture the bubble very quickly without puncturing the tissue underneath, and then you cut out the quadrants. Now, this was termed as a Descemet's bearing technique, that we are now bearing the Descemet's membrane, as you see below, on which we put a new cornea from which the Descemet's membrane has been removed. Now, while doing this operation, I, looking at it, found that there were certain clues that were telling me everything is not what is claimed to be. For example, the bare Descemet's membrane is a bit more resilient and tough. If there's a small perforation, it doesn't extend like a rip and curl up like we know the Descemet's membrane does. There's something else going on over here. And when I do a penetrating graft, as I emerge my needle from the depths, and I don't go through the descemet, I try to emerge my needle anterior to the descemet, I see that the needle is coming out and there's a nice sharp edge. And we always attributed this sharp edge to the descemet's membrane. So that, just bear that in mind as we go through the next two slides. Now, when we peel off the descemet's membrane from the stroma in a Dahlke operation, so we are removing the donor descemet, it peels off very smoothly. It comes out very nicely all the way through, as you can see. And this is also relevant. So I will come back to this point in the next couple of slides, like I said with the previous slide. Now, imagine that we have taken this descemet off and we are going to transplant this cornea in the Dahlke operation. Now, here I am putting my needle in deep and I'm still seeing that edge. Now, if that edge was due to the descemet's membrane, I've just removed it. Why should I still see the edge? There's something else producing that edge effect. So that was another clue. And then this was very interesting, that sometimes when the bubble did not reach the edge of the trifine mark, you have to separate the deep stroma from the anterior surface of the descemets. Now, if I'm separating it, I'm seeing these strands that are extending from the posterior stroma to the anterior surface of the descemets. Now, I just showed you when I strip the descemets from the stroma, it is so easy to peel it off. Why should there be collagen strands between the deep stroma and the anterior surface of the descemets when I'm trying to peel the stroma off? So that also didn't make sense. So put all this together, we said this hypothesis that there exists a distinct layer in the posterior stroma that is different from the descemet's membrane. And in the big bubble Dahlke technique, it is this plane that we are cleaving, not really the anterior surface of the descemet's membrane from the deep stroma. And that was the hypothesis. And we call that the layer as the pre-Descemet's layer. And we went on to do a series of experiments to show that that was the case. And the very first data on this was presented at the annual Congress of the Royal College of Ophthalmologists in 2007, so almost 6 years before the paper was published, and at the Italian Ocular Surface Society, which is now the SIXO, in Lecce in also 2007 June. At that time, we had called it the pre-Descemet stromal layer. Now, these were the experiments. So this is the human cornea, and you inject air. Notice how the air moves in a circular manner. Then it comes in, small bubbles form, and it lifts it up in a big bubble. This bubble rarely goes beyond 8.5, 9 millimeters maximum. And this bubble was centrally located. And you can see examples of this bubble over here. And this is in a much younger eye. And we call this the type 1 bubble. What it showed was that the air lifts this layer off, but it doesn't go through this layer. So this layer is impervious to air. Similarly, sometimes we found that when you inject air, notice that circular movement, then this bubble forms at the periphery and goes across the entire surface of the back of the cornea. And here again, another example, circular movement of air at the periphery, filling in. And here there's a bubble forming, here there's another bubble forming. The two of them merge together to extend across the entire posterior surface. And this too is impervious to air. The descemet's membrane does not allow air to go through it. We call this the type 2 bubble. And sometimes we found, which was not very common, that both the bubbles appeared together. There's a central, fairly large type 1 bubble, and then this horseshoe-shaped bubble going to the periphery, which is a type 2 component. So we call this the mixed bubble. Nowadays, some people call it the type 3 bubble. It's not a different type. It's a combination of the two. Here again, a central type 1 bubble, and at the periphery, there's a crescent shape like this, a little bit of air anterior to descemets called the type 2 bubble. So this is a combination. So together, it's the mixed bubble. So that's what we said is happening. And then really, this was the eureka moment. We created a type 1 bubble, and I was able to peel off the entire descemet's membrane, and the bubble was still there. So what is this layer which is holding the air underneath? This layer is consistently separated when we create this kind of bubble in the human cornea. And that is the layer that we were talking about. Now, just a still picture of the same process. Now, when the type 1 and 2 bubbles were created and we looked at the mixed bubbles, it was very interesting because clinically, when we were seeing mixed bubbles and people were seeing this clear margin bubble and the white margin bubble and a double bubble, and they were attributing the double bubble, which we now know is the mixed bubble, to a split between the banded and non-banded zones of the Descemet's membrane. It's in the textbooks, it's in the paper. But here's what happens if you get a type 2 bubble and you try to peel it off, the bubble completely collapses. So there is no pre-Descemet's layer underneath. You can see the whole Descemet's membrane has been peeled off. And that cornea now, where you've taken the Descemet's membrane away from, you can inject air in it and and you can still create the type 1 bubble. That layer, the pre-Descemet's layer is there, so it will still lift off to create that bubble. So you don't need the Descemet's membrane to create this bubble of the pre-Descemet's layer. So that was another point proven that, again, that they are two separate layers. And when we took the Descemet's membrane from the type 1 bubble, from the type 2 bubble, or from the mixed bubble, and you do histology, and this is low power electron microscopy, you can see there is never a split between the banded and non-banded zones. So what was in the literature was actually incorrect. And we know now that the mixed bubble was not due to the split, but it is due to the two bubbles happening simultaneously. We went on to characterize this in many different ways, and you can see by scanning electron microscopy on the top right, you see the Descemet's membrane has been peeled off, and underneath the very smooth surface of the posterior part of the pre-Descemet's layer. On the side view, you can see that's the edge of the pre-Descemet's layer, that's the posterior surface, and that is the Descemet's membrane that has been peeled off. We characterize this in many ways. You can see scanning EM pictures, one side and the other side. This is a nice bubble. You can see this bubble, that's the layer, pre-Descemet's layer. The Descemet's have been peeled off, and you can see these little strands that are extending at the periphery. If you look at the posterior stroma from which the pre-Descemet's layer is being peeled off, you see these tiny little fenestrations from which the air comes out in the form of tiny bubbles posterior to the pre-Descemet's layer and lifts it off. And this is very high power electron microscopy. You can see this is a scanning EM. That's the pre-Descemet's layer. That's the edge of the air bubble, and that's the stroma anterior to it, which is being peeled off. So we looked at this and we published that first paper on the pre-Descemet's layer. And at that time, we also postulated that it probably has a role in Descemet's detachment, in pre-Descemet's, in descemetocele and in acute hydrops and so forth. So we'll come to that in a minute. But that paper, like anything new that is published, evokes four kinds of responses. A lot of people said, and some of them were very genuine concerns and questions people had. First response is, it cannot be true. Second response is, it has been described before. Third response, which I also experienced, was, I have described it before. People claim they had described it before. And then the final, which is fortunately the case with the majority of the people, they acknowledge it. They say, well, let us test it out. They do more experiments and the evidence builds. And over time, it is either discarded or the new discovery is accepted and comes into common practice. That's the process through which any new discovery goes, and this discovery of the layer was no exception. So there was an editorial in Ophthalmology soon after this was published saying it has been described before. There is nothing new under the sun. I responded to that editorial saying, yes, there is nothing new under the sun, but the sun is constantly shining new light on old objects. We'll come to this again a little later. Here you can see on transmission electron microscopy, that's the layer, that's the big bubble from which the endothelium has been stripped off. This was the picture that was published in many, many newspapers at least in the UK and other parts of the world. This is a scanning electron microscope. You can see that's the layer with the type 1 bubble. It comes only to 8 or 9 millimeters. Beyond that, it is firmly attached to the stroma. That's the Descemet's membrane that has been cut. So you can see that this layer extends all the way to the periphery, but the bubble does not extend all the way to the periphery because of this firm attachments. Now, some people said, oh, it's random separation of the stroma. It's not really a true layer. So we did this experiment with my colleagues, Mastro Pasqua and Mario Nubili in Katy. We took a human cornea, we took a bunch of samples of them, we mounted it upside down on an artificial chamber and did phototherapeutic keratectomy to ablate the thickness of the layer. And we said if it is random separation, then we should get another bubble. So we now inject air, and what you see is that air escapes through multiple little holes there, but no layer, no bubble is formed. So clearly, once you ablate this layer, it's gone, and it is a distinct layer. It is not a random separation of the stroma. So genuine question, that the genuine answer. And on histology, you could see that where the arrow is, that's where the pre-Descemet layer is still there, but in the center, it was ablated by the phototherapeutic keratectomy. Then people said, oh, all your eyes were 70 or 60 plus in age. What about children? Then this was this 9-year-old child who had a DALC operation, but the cornea never cleared after the DALC operation. And OCT showed that, yes, there was a type 1 bubble, but actually it was a mixed bubble. And the type 2 component had ruptured and the Descemet's membrane had torn and curled. So this cornea did not have an endothelial lining in the center because the Descemet's had torn, and therefore the cornea had never cleared. Later, penetrating keratoplasty was done, and on histology, we could see that the pre-Descemet's layer was there, distinct from the Descemet's membrane. So yes, it was present in children, and that question was answered. Even more, when Amar Agarwal and I did some experiments with very young eyes. These were children's eyes, which one aged 3 weeks old, one aged 1, 4, and the 9 years one I showed you. And in all of these eyes, you can create a type 1 bubble and you can show that the pre-Descemet's layer is there. So the answer was yes, it is present in children as well. And then they said, oh, if it is present, then why can't we see it on OCT? And the answer was that the OCT resolution wasn't good enough, but with high resolution OCT, Bezeva et al. From Waterloo in Canada published this paper, and they showed quite clearly, just as we had said in our original paper, the facts we had said that this layer separates beyond the last row of keratocytes. And what they did is, this is the area we're looking at, this red square. In high power, you can see the Descemet's membrane always gives you a double contour line. You have 2 lines. Then the last row of keratocytes is by the red arrows, and in between is the pre-Descemet's layer. This is in the living human eye. And look at the corresponding histology. Just as we had said in our first paper, they've confirmed that, saying that there's the last row of keratocytes, there's the pre-Descemet's layer, and that's the Descemet's membrane with the endothelium, which you can see corresponds to the ultra-high resolution OCT picture. And now others have gone and shown that in the living eye, yeah, you can see it on OCT. Is it present in animals? Yes, it is. We did experiment on, this is all published work in Scientific American. We had these spaniels from which the eyes were taken, and they have larger eyes, and we got only type 1 bubbles. We never got type 2 bubbles in these dogs, but it was consistently shown that yes, in animals, and we've done it anecdotally in a couple of other species, and it's there, but it's interesting how the bubbles behave. Looking at this in more detail, we went on to do some further experiments which have very significant clinical impact or translational impact. So this layer stretches when you put the air in a big bubble, and it comes back to its normal shape. So clearly, it has a lot of elasticity. So we tried to look at the elastin content of this layer and other parts of the cornea. And you can see in this graph, by extracting the proteins and measuring elastin, the pre-Descemet's layer has the highest content of elastin, equal to the trabecular meshwork, but it is less in the Descemet's membrane and even less in the central peripheral cornea and in the sclera. So if you take the pre-Descemet's layer and stain for elastin, green is elastin, the whole layer stains for elastin. And this is very novel finding at the time. But if you stain the Descemet's membrane, only the top band corresponding to the banded layer stains with elastin. The rest of the cornea does not stain with elastin. The rest of the Descemet does not stain with elastin. This is the wall of a type 1 bubble. The pre-Descemet's layer stains. There's the anterior band on the Descemet's membrane, but most of the Descemet's membrane doesn't stain. And here again, so these are all isolated samples of the wall of the big bubble. So again, you can see this was essentially be what we now know as PDEC tissue, and we'll come to that when we look at the surgery. But here again, you can see the pre-Descemet's layer, the anterior band of the Descemet's here. But what happens if you look at the whole cornea without stripping the layers? And this is a very nice example. You can see the endothelium, the Descemet's membrane, no elastin stain, this band corresponding to the banded zone. The interfacial matrix is dark, and then the pre-Descemet's layer uniformly staining with elastin, and the rest of the cornea has very little elastin. So this was histological evidence showing that biochemically this layer is different. Structurally, it is different from the rest of the cornea. And that also explains why, if you take the PDEC tissue, and like I said, we'll have a quick word on that later. But if you look at the pre-Descemet's layer and Descemet's membrane together, they scroll to that extent, about grade 2 scroll. If you separate carefully from this tissue, the pre-Descemet's layer and the Descemet's membrane, the Descemet's membrane scrolls the most, the pre-Descemet's membrane scrolls the least, and the two together scroll somewhere in between. Now look at this finding. This layer has the maximum elastin, but it scrolls the least. This layer has much less elastin, but it is uniquely distributed, the Descemet's membrane, and it scrolls the most. And now, why does the Descemet's membrane always scroll with the endothelial cells outside? And there were different explanations given. One was that it is the content, the elastin, which is true, but it's not just the elastin, because then the pre-Descemet's layer would also scroll like that. It is the distribution of elastin. So what we did is we took a sample of the scroll Descemet's membrane, we put elastase enzyme and let it unscroll, and spontaneously it unscrolls. You don't have to tap it or manipulate it. You put the elastase, over time it becomes flat, like you see over in the left middle picture, and you do histology of that, and you can see that that band of elastin is completely fragmented as against the controls, which stay as they are. The other explanation given was that it is due to the swollen endothelial cells that post-mortem, when they swell, they will push the Descemet's to scroll with the endothelial cells outside. So we did this experiment where we took dispase enzyme, very lightly treated the Descemet's membrane, and we washed away all the endothelial cells, put an F mark to show which way was the endothelial surface, and it still scrolls exactly the same as you see in this example. So clearly, the swollen endothelial cells have no role to play in the scrolling of the Descemet's membrane, but that's in the literature. People have given this explanation as the reason, but the evidence was not there. In fact, the evidence is completely the opposite. Now, let me try to explain this a little with this small cartoon that we made ourselves. So if you look at this rubber band, you stretch it and you leave it. It comes back, you stretch it and you leave it. So that's elasticity of the pre-Descemet's layer. Now, if I hold this, you put air, like the big bubble, it goes down, the air comes out, it goes up, okay? So that's what's happening with the pre-Descemet's layer. Now, imagine this is that band. So you put some glue on this rubber band and you stick it to the strip of paper. Now, this is the anterior elastin layer on the Descemet's membrane corresponding to the banded zone. Now look what happens when, because of that distribution of the elastin, when I leave it, when it's cut, then that whole paper will strip with the endothelial surface, the blue surface outside. So that is why we get the scrolling of the Descemet's membrane. And as we showed that if you destroy this elastin with elastase, then the scrolling doesn't happen, or if it's scrolled, it unscrolls. So the explanation is there for something we take for granted in that layer. Now, God didn't make this layer for us to do DMEC. Why did he put elastin there? The physiological role would be, and this is my explanation for it, that when the cornea is concave at the back, the Descemet's membrane has to be firmly attached to it so that it can function and to deturgest the cornea. And there's no physical attachment between them. There's just an interfacial matrix. So what this Descemet's elastin does, it tries to make the Descemet scroll like this. All the time, the forces are against the back of the cornea, holding it in place. And that's why you find when the elastin degrades with age, then the Descemet's detachment becomes easier. So discovery of this layer has improved our understanding of DALK through novel understanding of posterior corneal anatomy. It has informed intraoperative interventions to make DALK safer. And it has also introduced paradigm shifts in our age-old concepts of corneal pathology. So this is what we will look at next, how it has impacted surgery and how it has impacted pathology. All of this information is in these two references. One is a book chapter on DALK by Albert Jacobiak. That's a little old compared to this review article with some new information in Progress in Retina and Eye Research published in 2023. So when we inject air and we do this DALK operation, I've shown you this video before, you get this bubble and you can take it off and you do the DALK as before. So we'll skip the rest of this video, go on to the next. When you're doing air injection and you see that clear bubble at the periphery going all the way, there is no white line, and you can see that that's the type 2 bubble, exactly simulating what we had done with the ex vivo experiments. And then question is, if the pre-Descemet's layer is impervious to air, how does air go through it to create a type 2 bubble? So air has to reach the anterior surface of the Descemet's membrane to create a type 2 bubble. But the clue was in the fact that the bubble always started at the periphery. So what we did was, when we were injecting and the bubble was forming, we marked the point where the bubble was forming. Then we let the bubble form and we stripped the Descemet's membrane away from that point that we had marked. And we looked underneath with scanning electron microscopy to see what is different over there, that air is coming through. And what we were able to show is that there are these clusters of holes, single or multiple, at the periphery of the pre-Descemet's layer, and they are about 15 in minute distributed randomly at the periphery, and they are 50 to 60 microns in size with the mean of 20 microns. And that's why the air will escape through the periphery. So let me illustrate that in this high-power scanning electron micrograph of the human peripheral cornea. Another important point, which we'll come back to also, is that this pre-Descemet's layer, the Descemet's has been removed, actually at the periphery becomes the trabecular meshwork. The trabecular meshwork is in continuation, is one structure with the pre-Descemet's layer. At the periphery, it opens up. Now, where it is opening up, you will see these fenestrations, single or multiple holes, and then it's actually the holes of the trabecular meshwork. If the Descemet's membrane is attached centrally to these holes, air will escape through these holes and go into the anterior chamber, and we see that quite often in DALK. But if the Descemet's membrane is attached at the periphery to those holes, then air will escape through those holes and go anterior to the Descemet's membrane and create a type 2 bubble. That was an explanation of why these bubbles were different. And this is a nice video sent to me by Professor Tariq Katamesh, where he has to illustrate a mixed bubble. So there was a bubble of air underneath. He punches the pre-Descemet layer to release the air between the pre-Descemet layer and the Descemet membrane. So we now know what's happening, so we can deal with it, and we can show exactly what the mechanism is for the creation and the formation of different types of bubbles. Now, how is it clinically significant intraoperatively? Imagine if you got a mixed bubble like this. There's a large type 2 component, and then this is type 1 in the middle. If you're going to do paracentesis in this eye, you're going to puncture the Descemet. So you don't want to do a paracentesis unless you have released the air from the top and everything's gone back. But if it is like this, you will not put your paracentesis needle from, you will put it from this side where the type 2 component is missing, but you will not put it from this side where there's a type 2 component, because you're more likely to hit the type 2 bubble. So your paracentesis needle or knife should avoid where the Descemet membrane type 2 bubble is. And this is, so look at this eye, nice bubble was created, and I'm trying to trim the edge, and look what happens. We didn't know then that there's a type 1 and type 2 bubble, and the whole Descemet membrane ruptures. And you can see, so this Descemet membrane is holding the entire intraocular pressure, which normally is held by the cornea of 500 microns. Here you have a 20 micron or less structure holding the pressure, and you touch it. It's like an inflated balloon. You touch it with a pin, it will burst on you. Look at the bottom video now. Before I started, let me explain. So the doctor of a friend, Kana Ramesh, has lent me this video. So a bubble was formed, and the donor Descemet membrane was removed. The donor cornea has been put on the bubble, and for some reason, the surgeon holds the conjunctiva and pulls the eye to one place. That pressure is enough to do what you'll see next. So just pulls it to one, and that whole Descemet membrane bursts. At that time, we did not know the difference between type 2 and type 1 bubbles. Now we can avoid these disasters. You can have a very successful DALK with a type 2 bubble if you constantly deflate the aqueous, keep taking the aqueous out through paracentesis to keep the Descemet membrane flaccid, to keep the pressure low and not tense. If the Descemet is not tense, it will not rupture. If it is flaccid, you can do your manipulations, you can do your suturing. If you touch it, it doesn't. So that release of aqueous you have to do every minute or every 2 minutes. It's not that you can do it once and finish the operation, because the aqueous keeps forming and it can be under pressure. So we've learned a lesson, and we have made our surgery more safe. Again, these strands of collagen that extend from the periphery to the center, and we can show that here, again, stripping off along the last row of keratocytes. This tells us that we cannot make too large a trephination, because the attachment of the pre-Descemet layer to the stroma is very strong at the periphery. So if your trephination is too large or it is eccentric, then you will encounter an area where you have these strands which have to be physically separated. These strands, which are broken in the center, they're bits of collagen on the layer that always remain. And you can see by looking at it under the operating microscope, when there's a rough appearance, that's the anterior surface of the pre-Descemet layer, therefore a type 1 bubble. Where there is a very smooth appearance like this, no rough appearance, no strands, that's a type 2 bubble, and that is the anterior surface of the Descemet membrane. Fortunately, 85% of the time, the bubble is type 1, and about 15% is either a mixed or a type 2. And you can get a good outcome with both the bubbles, but with this type 2, you have to take more precautions like I showed you. Another interesting thing was, in the experiment, we found that you keep pushing air, and it takes a pressure of about 700 millimeters of mercury to rupture the bubble. And that was an indication of how strong the pre-Descemet layer is. And here you take away the Descemet membrane, and you can see that's the layer. And that layer is very translucent, resilient, elastic. And if you pull it, you can see the striae extending all the way to the periphery. So again, the point that the layer The pre-Descemet layer goes all the way to the periphery, but the bubble only goes in the center. So based on this, my colleague Ahmed Zaki from Cairo said he was doing DALK and suddenly found there's a dense cataract. So he was able to do the whole cataract operation with lens implant, and in one patient even had to do anterior vitrectomy. All of that under the layer, you keep it, the fluid going, keep the chamber formed. You can do all of that and you put the lens in. So he did a series of cases we published together as the DALK triple operation, where you can do this deep anterior lamellar keratoplasty, cataract extraction by phaco and implant in these patients. So that's the DALK triple, and that there were a few cases of that. The other important thing is that if you get a type 1 bubble and it tears or ruptures, it is not the end of the world. You can still complete DALK successfully. As in this case, you will see there's a tear over there. I'm putting my cannula in that. You can see that tear. That is not, but if that happened in a type 2 bubble in the Descemet, it would spread and rip. But in a type 1, you can complete the operation successfully. And post-operatively, all we saw was a little wrinkle, a fold in the pre-Descemet layer, but the outcome was pretty good. When you're getting a type 1 bubble like this, it suddenly burst in the eye. So we know that something has gone wrong. You don't have to abandon the operation and do a PK. The bit of bubble is still trapped over there, bit of air, because we saw the white ring. So we know the bubble had formed. So you can open that bubble. You can access that plane that the air was created. Use viscoelastic, very, very safe to use viscoelastic, just needs to be washed away better, and you can then complete the operation successfully. You see it is leaking over there, aqueous when I touch, but the aqueous is leaking, and that's the type 2 component that had formed and ruptured. And you can see post-operatively, there's just that little tear over there, which was underneath the host rim. So all these things are now making sense. Excuse me. And we are able to understand what's going on and how to deal with it. I showed you again this little child where there was the same thing happen. The type 2 component has ruptured. Now, also look at this. The air has gone straight in the air. So I punctured Descemet. So not to give up, go to another site. You create a small type 1. That's good enough, because I know there's a cleavage plane between the posterior stroma and the pre-Descemet layer. So I put some viscoelastic, I find that cleavage plane, and with a spatula, you can completely separate the whole and convert it exactly as though it was a type 1 bubble and get the same outcome. So even when you don't get a type 1 bubble or a type 2 bubble, when you do manual dissection, you can reach that plane, put a spatula in, and separate it all. So that's what we do so many times. This is another application of the knowledge. Rishi Swarup said they tried a visco bubble, they didn't succeed. So you can simply peel off the whole stroma from the pre-Descemet layer. And that's called the peeling technique, because there is this clear cleavage plane between the anterior surface of the pre-Descemet layer and the posterior stroma, which we can now exploit surgically. And then another example to show you how this knowledge helps us. So I'm doing a DALK. I didn't get a bubble, so I'm doing manual dissection, hydration, and carefully I am separating the anterior stroma. But then I noticed that I've got something different over here. And that there is a different edge. And that's the pre-Descemet layer. And I can put my spatula underneath between it and the Descemets, because here the Descemet is exposed. But I know that I had cut the pre-Descemet layer there, but I then exploit that cleavage plane anterior to the pre-Descemet layer and come from the other side. I finish the complete excision of the deep stroma. This is like a mixed bubble where the pre-Descemet layer was cut. That's the edge of the pre-Descemet in that sector, and we were able to complete the operation successfully. So when we see these things, we know what's happening. Our understanding has improved considerably. Another surgical application is that if you inject air and create a type 1 bubble, you can take the air away and you can refine that tissue and use it for endothelial keratoplasty. Again, this was something we had anecdotally done with the video, ex vivo. And in the very first paper in 2013, we had 13-14 explained this technique of endothelial keratoplasty using the wall of a type 1 bubble because it scrolls much less, and that is now established as the PDEC operation. So you can see here, this is the bubble in the donor eye. You can refine it or you can cut it. You can load it. This is on a lens injector. It also scrolls with the endothelium outside, like I showed you earlier. You can put it in an injector, put it in an eye from which you've taken the Descemets, use any way you want to unscroll it. It's much easier to unscroll, and that is the operation of Pre-Descemet's endothelial keratoplasty or PDEC. It's a direct innovation of the discovery of this layer, and these are some examples of PDEC. You can see day 1, 2 months later, there is absolutely no difference in the visual recovery and the clarity of the cornea between DMEC and PDEC. For that matter, even ultra-thin DSEC gives you the same results. Now, let's look at the pathology before we finish. And this is where it has really come into it. So this has really shaken our concepts of long standing, some even more than 100 years ago. So 98 years ago, the definition of Descemet's membrane detachment was that it is a detachment of Descemet's membrane by Samuels et al. We completely changed that concept, because just like you have the 3 types of bubbles, you get 3 types of detachment. A type 1 detachment is like a type 1 bubble. Now, these are eyes where no air is being injected, nothing of that sort. These are patients coming to the clinic. And this is a Pre-Descemet's layer with the Descemet's membrane endothelium that is detached. It is like a chord of a circle, and it is a straight line. Type 2 is a double contour line. The Descemet's membrane gives you 2 lines in the OCT, and it's wavy. It's not a straight line. And sometimes that can be torn, and it will scroll with the endothelial cells outside, and that will be called a regmatogenous Descemet's membrane detachment. And look at the patient 3 here. There is a double detached. Pre-Descemet's layer is detached, straight line, and the wavy Descemet's membrane detachment. So like a mixed bubble. So the Pre-Descemet's and Descemet's have separated from the posterior cornea, but also separated from each other. Type 3 detachment, exactly like the 3 bubbles, and they behave differently, and their treatment is different. So this is a patient, chronic edema, post-cataract surgery. On slit lamp, you can see straight line in the middle corresponding to the area of the type 1 bubble. OCT, straight line. This is a type 1 detachment. It is not Descemet's detachment only. It is Descemet's and Pre-Descemet's layer detached together. Of course, the Descemet's detached, so we call it a type 1 detachment. After, again, let me show you histological evidence we were able to collect. So type 1 detachment, and you can see on histology, the Pre-Descemet's layer has separated the Descemet's membrane, and the endothelium is over there. Here's another example. In a failed graft, there is a Pre-Descemet's layer detachment. And if you see this strand on OCT between posterior stroma and the Pre-Descemet's layer, then you're guaranteed that this is a type 1 Descemet's membrane detachment. This is a Pre-Descemet's layer, because there are no strands, like I said, between the posterior cornea and the Descemet's membrane. And on histology, we could see that straight line. And with time, the keratocytes will migrate into the layer. It becomes variable in thickness, and it shrinks. Therefore, if you just put air inside, put it back, it doesn't work. You have to release the tension as well. Again, another example here of a mixed detachment. Here you can see there are 2 lines. Pre-Descemet's layer, Descemet's membrane, 2 lines. So different colleagues have contributed. We're all co-authors on this paper in the AJO. And here you can see at the periphery of the detached area, it is type 1. As you're moving centrally, it's become mixed. And as you come right in the middle, there's a type 1 component there, and the type 2 has torn and scrolled. So it is a regmatogenous mixed or a type 3 Descemet's membrane detachment. Here's another example of that strand extending between deep stroma and anterior surface of the detached Descemet's, which is the Pre-Descemet's layer. This was a very nice clinical example, again, of a patient. I won't go into the whole detail. It's published. Advanced Murren's ulcer, and you can see eventually treated, and a spontaneous type 1 detachment was there. A slit lamp, that straight line. So when we are seeing these things, we know now that we are seeing different things of when we see detachments, they're not all the same. What about acute hydrops? For more than 100 years, we have been taught and we have been teaching our students acute eye drops is due to rupture of the Descemet's membrane in eye with keratoconus. Not true. Rupture of the Descemet's membrane alone does not cause acute eye drops. You need to rupture the Descemet's membrane and the pre-Descemet layer. Again, this is something we published in the very first paper. All these concepts were there because we had seen anecdotally cases like this. This was a patient who had acute eye drops. PK, penetrating keratoplasty, was done, and on histology, we could see that both the layers had ruptured. And then when you do now OCT on all your patients with acute eye drops, you will see, sometimes you will see, we are able to see the rupture always, but you'll see the separation of the two layers and the fluid vacuoles in the stroma. That led two other workers to make some very important supportive comments. One of them was Professor Murain from France, who said that if this is true, then what he did was put air in the eye with acute eye drops, marked the ends of the tear, and he said by just approximating the torn pre-Descemet's layer together, he was able to induce rapid resolution of acute eye drops. So he said the fact that the pre-Descemet sutures can be also effective could be explained by the existence of the Duer's layer. Acute corneal eye drops could be due to tear in the layer, etc., etc. That was him. And then Gerrit Mellers, who initially was a bit skeptical about the layer, published this paper. He says, are Descemet's membrane ruptures the root cause of corneal eye drops in keratoconus? He questioned this because he referred to our previous three papers on this fact that they are not. And he was doing Bowman's membrane transplant. And while creating a pocket in the mid-stroma to do Bowman's membrane transplant, sometimes there was Descemet's membrane detachment. There was no acute eye drops. But in the cases where, while creating the pocket, he inadvertently perforated posteriorly through the pre-Descemet layer, he got acute eye drops. So he said, you need a rupture in both these layers to get acute eye drops. But the story didn't end there, because when we read this paper, we were very pleased that, yes, he's corroborated in a living experiment what we had shown with evidence on histology. But we went on to do this small little experiment. You take a human eye and you make a slit in the back through endothelium and Descemet's membrane. You mount it on an anterior chamber, artificial anterior chamber, and you push the pressure up to 80 or whatever you want. You don't get acute eye drops. So split in the two layers, even with raised pressure, does not cause acute eye drops. So what's going on? Acute eye drops results from a tear in Descemet's membrane and the pre-Descemet layer in the context of the abnormal collagen and proteoglycan matrix of the keratoconus cornea. So it's the whole pathology, not just the tear, that leads to this sudden hydration, because it does not happen in normal corneas. And what about descemetocele? Now, this is very interesting. We have seen descemetoceles forever. We know some of them burst readily, some of them don't. And to cut a long story short, they all look the same, but there are three types of descemetoceles, not corresponding to bubbles, all three. They're slightly different. Type 1 is exactly like the type 1 bubble, where you have the Descemet's membrane and the pre-Descemet's layer on it. Type 2 is where it's only the Descemet's membrane, like a type 2 bubble. But the type 3 is where there is Descemet's membrane, pre-Descemet's layer, and a bit of stroma. And all the three will look like they are bubbles on the anterior surface of the cornea. But on OCT, you can see there, this is only Descemet's membrane, that's Descemet's membrane with the pre-Descemet's layer. But this very interesting case recently reported an eye opened by my fellow, who's now an associate professor at Birmingham. They had done a DSEC on a patient. The patient got infected. And everybody will say this is a descemetocele. But this patient did not have a Descemet's, because the Descemet's was removed from the recipient eye in the DSEC operation, and the DSEC graft was put underneath. So why is there still a descemetocele-like appearance? This is what's called a pre-descemetocele. It was the protrusion of the pre-Descemet's layer, not the Descemet's membrane, because there wasn't a Descemet's membrane. And of course, it went on, after they treated it, the stroma remodeled and it healed. And that was the area of the pre-descemetocele. So the clear distinction in the pathology of desmetocele that is happening, which we now understand because of the knowledge of this layer. Again, in infectious keratitis, we have published this 2 or 3 cases in our paper in the Progress in Retina and Eye Research. When you get deep fungal infection, you get a type 1 detachment with inflammatory debris inside it. It's a very good biomarker for fungal infection. It can happen in any deep infections, but if you see a type 1 detachment with inflammatory collection between it and the stroma, think of fungus. I don't know how many of you have seen an intracorneal hypopyon. I have, and that is a well-defined entity. The separation is in the area of a type 1 bubble. We now know, of course, these were published long back. The hypopyon is in mid-cornea. You see, that's the limbus. So that's not where the hypopyon is. It's a separation. It's a type 1 separation, as you see on, I've just put the OCT vertical. So that's a type 1 detachment, and the hypopyon level is where the pus is accumulated. So intracorneal hypopyons, we now know, is because of pus accumulating in the cleavage plane afforded by the pre-Descemet's layer and the Descemet's membrane. And there's also a Lewis dystrophy. This is a biclonal gammopathy syndrome where you get the deposition exactly in the pre-Descemet's layer at the back of the cornea. So there are many pathological conditions where we are beginning to make some sense of. And I'm just going to finally go through one concept which is new in the sense that the implication of the concept is new. The concept's very old. It was our second paper on this layer, that the pre-Descemet's layer continues as a trabecular meshwork, which I told you before. And
Very soon
sorry,
after the
there,
discovery of this
let me
layer,
switch this off.
we were able to publish
That's the evidence I showed you.
it showed
This slide shouldn't have been here, but what we were able to show is that, sorry, let me
incidence
just
of glaucoma after full thickness corneal
let this run.
transplant, where the pre-Descemet's layer is cut, the incidence of glaucoma is higher than after deep anterior lamellar keratoplasty, where it is well known that.
Let me just get this right down. Sorry about this. So we know that the incidence of glaucoma is higher after penetrating keratoplasty than after DALC. And the difference is that the pre-Descemet's layer is cut in DALC, is not cut in DALC, but is cut in penetrating keratoplasty. And we know that the pre-Descemet's layer continues as a trabecular meshwork. So we have this ex vivo model of the human cornea perfusion model. It is mounted in an artificial anterior chamber and is perfused with culture medium coming from the top on a balance. So you can actually measure the aqueous outflow in this model. So if you take the human cornea and you cut it 180 degrees corresponding to an 8 millimeter trephination, and then you put this on the model and you measure, you will find that the outflow of aqueous in these eyes is significantly less than in eyes where you have not made the cut. So we have not cut the stroma. We have only cut the pre-Descemet's layer. Cutting the pre-Descemet's layer does impact on the pressure dynamics of those eyes. And this is a paper recently published in Frontiers in Ophthalmology. But we know also that when biomechanical stress is altered, protein expression and gene expression alters. So we went on to look at the different proteins, and we found the fibrotic proteins like fibronectin, alpha smooth muscle actin, myosin, collagen 4 and vimetin. All of these are implicated in glaucoma, are increased in the trabecular meshwork of the side where it is cut compared to the side where it's not cut, and in comparison with the normal controls. So that is this paper here, just I think is published earlier this year, 2026 paper. And then we were also able to show the MMPs and all by zymography and the myosin expression. So we did the protein expression as well, not just by immunohistology. So the concept or the hypothesis is that altered biomechanics by incising the pre-Descemet layer leads to alteration and expression of critical fibrotic proteins that reduces the aqueous outflow, and that is probably going to impact on pressure in the eyes. So with time and knowledge, like I said, new applications of this layer will emerge. So to summarize, the discovery of the pre-Descemet layer has improved our understanding of posterior corneal anatomy, of deep anterior lamellar keratoplasty and endothelial keratoplasty, and made this operation safer. It has helped in the innovation of procedures and techniques to deal with and overcome intraoperative challenges. It has shed new light on corneal pathology, concepts that we had taken for granted for many years, we know now are not entirely correct, and particularly with Descemet membrane detachment, acute eye drops, and a few others. It has led to the innovation of 3 new procedures. One is suture management of acute eye drops, dark triple, and the pre-Descemet endothelial keratoplasty procedure. Understanding what is happening will give the surgeon more confidence in performing DALK and dealing with the complications. If we know why we do what we do, we can do it better or find a better way to do it. This is the paper I was referring to, which is a good reference paper if people want to go back and look at some of the evidence behind what I have presented. Thank you very much.
Thank you so much. Wow. Oh, Harminder, sir, what a presentation. Really superb lecture, Professor. That is extraordinary. And now, fellow ophthalmologists, now we are approaching the question and answer session. Let me read the first question, Professor. Sir, thanks for the great lecture. Please explain once more, what is the quickest and more reliable intraoperative cue to differentiate a type 1 from a type 2 big bubble before making the incision?
So you cannot predict which bubble you're going to get. You cannot predetermine with a technique you will get this type or that type. That's not possible. But once you get it, you can tell which type it is. But if you look at the surface of the bubble that you've created when you've taken the stroma off, you will see very clearly that rough appearance, and you know this is a type 1. But you will know that even while it's being formed, when you see the white ring forming. If you're doing it manually, of course, you will not see the white ring, but you will be in that plane. Very easily, your spatula will move. You know you're in the right plane. So that's the difference. We know clinically now, though, that people who have deep stromal scars are the ones where you're more likely to get a type 2 bubble, because there the pre-Descemet layers is also scarred and attached to the stroma. And if the inflammation was excessive or prolonged, then air is more likely to go posterior to the pre-Descemet layer, anterior to the Descemet membrane, give you a type 2 bubble. That we have known by clinical experience, and that's been published.
All right. Thank you so much, Professor. The next question, great lecture, Professor. In challenging scarred corneas, what is your preferred technique to safely access the pre-Descemet plane without rupturing Duas layer?
So I use the big bubble technique as a default. In acute eye drops, do not try it, because there the healed pre-Descemet layer is weak. It will rupture. So don't try the air technique. You can try the manual dissection technique. But otherwise, in all dystrophies, in keratoconus, and in corneal scars, I will try with the big bubble technique. If I get a type 1, fine. If most of the times that's what you will get. If you get a type 2, you know how to manage it, and I'm confident now, so I don't mind. But if I don't get a bubble, and that will happen from time to time, then I have a different strategy. It's a completely different lecture on how to still achieve good DALK when you don't get a bubble. And my technique is to, because already there is air in the stroma, I then hydrate with balanced salt solution, make it really thick you've got a mixture of air and water. Then with a blade cut along the trifine mark, And I keep cutting because when I see the air bubbles, I know I'm not deep enough, because when air bubbles are coming, they are all in the stroma. So I'll keep cutting, and then when I see a dark line, and I know I'm deep enough, and then just peel it off, or with a crescent blade, shave it off. You will leave behind some stroma. If you leave behind less than 100 microns of stroma on the pre-Descemet layer, it doesn't matter to the outcome. You can, with OCT subsequently, you can hardly tell that there is, it's quite amazing how the OCT does not even show the interface if you've gone deep enough. So the mantra is go as deep as possible, try not to leave more than 100 microns of stroma. But in this technique, often you will find that you actually find the plane, and then you don't have to leave any stroma behind.
Okay, thank you so much, Professor. The next question, remarkable lecture. Professor, does the extreme tensile strength of Duas layer change your suturing depth preference when repairing the posterior stromal restoration? Thank you so much, Professor, sir.
So we have only measured the bursting pressure of that bubble. So you have, if you get a type 2 bubble, which is Descemet's membrane, you push the pressure less than 60, even 30, 40 millimeter, the Descemet's will rupture. So if you get a type 2 bubble, don't attempt cataract. But with the type 1 bubble, it is 700 millimeters of mercury. So it has got a lot of strength. And this is in the normal cornea. If you're doing it in a keratoconus cornea, it might be less, because already we have evidence, which I probably missed that slide, that in keratoconus, the elastin content of the pre-Descemet layer is gone. In fact, Keith Meek showed this first by serial scanning on-face electron microscopy, and we showed it by the immunostaining method, that in advanced keratoconus, there is no elastin in the pre-Descemet layer, so it has no tensile strength. It becomes very brittle, and that's why it can break and cause acute eye drops. So when you're doing, if you were going to measure the bursting pressure of the pre-Descemet layer in a keratoconus eye, it is likely to be less, but it's still strong enough to do manipulations like cataract operation, where the pressure hardly goes above 70, 80 millimeters of pressure mercury during the cataract operation. And you can even stitch it individually. You can catch the edges of the layer and you can stitch it to approximate them closer to each other like in acute eye drop. So it is easy to handle and easy to manipulate compared to the Descemet. But I don't have actual tensile strength measurements on it.
All right, thank you so much. The next question, given its peripheral extension to the trabecular meshwork, have you observed any clinical correlation between pre-Descemet pathology and steroid-induced glaucoma?
We haven't studied specific pre-Descemet's pathology. We haven't studied that. We know that there are certain dystrophies like cornea farinata, which is a pre-Descemet dystrophy, this Lewis dystrophy, which was said, which is a pre-Descemet dystrophy. There are certain conditions which might specifically affect, and like I mentioned, we know that in keratoconus, the pre-Descemet layer is specifically affected, and that could explain posterior keratoconus as an early sign of the development of keratoconus. But what impact they would have on the pressure or on the trabecular meshwork, we haven't yet. We've only just started looking at this. Like I said, we're doing these perfusion model studies, and clearly the concept that the trabecular meshwork is a triangular structure located at the periphery from the root of the iris to the Schwalbe's line, that is incorrect. And it does carry on as the pre-Descemet layer. And just to mention this, a lot of what we say and do today, if you go back in the literature, has been described. Now, Salzman, in a paper going back about 105 years, in a German paper, which we got a translation of, actually with light microscopy made the comment that the trabecular meshwork is inserted into the posterior cornea. So he had that idea that it's not just ending there, it is part of the cornea. So chances are, I mean, to answer your question, what happens to pressure and the trabecular meshwork in pre-Descemet layer pathology remains to be studied, but that there is a distinct relationship between the two, and therefore distinct relationship in the pathogenesis of glaucoma and high pressure. It is there. That is most likely, as we have already shown in that experiment.
Thank you so much for the explanation, Professor. The next question, please kindly explain once more during dysmetorrhexis in DMEC, how common is inadvertent stripping of Duas layer, and how does it affect endothelial graft adhesion? Thank you, Professor.
So if we're talking about the recipient, the host, not the donor. So but the donor also, it has certain implications. I'll tell you in a second. But in the recipient eye, the more the stromal edema, the more likely it is that the pre-Descemet layer has already been separated. And not just the amount of edema, but the duration. If the corneal decompensation and corneal edema has been there for months, and you now go and try and do a DMEC, you have to be very, very gentle, because even with the scoring, because it has happened to me and to my colleagues, to my juniors, many times that when you're stripping it off, the pre-Descemet layer also strips. So when the pre-Descemet layer strips, then you get strands of collagen hanging, and they can interfere with the opening of the Descemet membrane, or they can interfere with the attachment. So try best not to strip the pre-Descemet layer from the recipient eye. But there is a clear risk, as the individual who asked the question has mentioned, of doing that. In the donor eye, we've got another paper published, I think earlier this year or late last year, where we've shown that the Descemets, what were the peripheral attachments of the Descemet membrane, pre-Descemet layer, and stroma, and at the periphery, the pre-Descemet layer is firmly attached to the stroma, like I've said, but also the Descemet membrane is firmly attached to the pre-Descemet layer. So if you're trying to strip the Descemet membrane from the donor eye, don't go right to the trabecular meshwork. Come a little more central. We know that the more central you are, the more loosely is the attachment of the Descemets to the pre-Descemet layer. So if you are just inside the trabecular meshwork and you do your scoring there, you will bypass that area where it is firmly attached. So it's about a millimeter of an area where the Descemets is firmly attached. So if you're trying to separate there, you're more likely to tear your Descemets, or you will try to peel off the pre-Descemet layer as well. So avoid that. And if you want to do a PDEC, then peeling is not the answer. Air injection is the best answer. People have done with that. This is also important, the point. If you do visco bubble or viscoelastic creation of a type 2 bubble, if you are not deep enough, it's the only time you get an intrastromal bubble. The viscoelastic material injected in mid-stroma will rip the stroma, and it'll look exactly like a type 1 bubble on the table. And you think you've done it, but you go and do an OCT, you'll see a huge amount of posterior stroma is left behind. So the intrastromal bubble never happens with air, but it happens with viscoelastic. So that one has to be careful about.
Wow, fantastic. The next question, does preserving Duas layer offer better protection against long-term graft ectasia in DALK?
Yes, it does. So if you get a penetrating keratoplasty, a little injury and the eye bursts, and I said that in the beginning, that's a very weak graft host junction. If you get a type 1 bubble and the pre-Descemet layer is left behind and you've done a DALK, the eye is stronger, because that layer will be able to withstand more trauma, more injury, more rubbing, squeezing than, in theory, with a type 2 bubble or Descemet membrane only. So nobody's tested this, yes. Nobody's actually looked at how strong is type 1 bubble dark versus type 2 bubble dark. Even nobody has looked at what is the incidence of glaucoma. These are two ideas. In fact, the second one, where what is the incidence of glaucoma after a type 1 bubble versus a type 2 bubble in dark? Because in a type 2 bubble, we are cutting the pre-Descemet layer, just like in the penetrating keratoplasty. So is the pressure different? And if we were able to tell that apart, it will be further evidence that, yes, cutting the pre-Descemet layer in a type 2 bubble dark does cause the pressure to be higher, like in a PK. That evidence remains to be generated, but it's a simple one to do if you do a multi-center study. And happy to join with anybody who wants to do that.
Wow, okay, this
That I will be
opportunity.
weaker at. Yeah.
Okay, maybe this is the last question, Professor.
Sure.
If a microperforation occurs specifically in duas layer during dark, can we safely continue with lamellar surgery, or is
Yes.
conversion to PK not mandatory?
No, not mandatory at all. In fact, even if you get a big macro perforation, just carry on with the dark. The advantage is phenomenal, because for the rest of the patient's life, he's not going to get endothelial rejection, and it heals quite well. So if you get a perforation in a type 2 bubble, it is more difficult to manage because it extends quickly and it rolls. So you have to do it like a Descemet detachment. You have to treat it. And if you can do it, that's fine. But in a type 2 bubble where the pre-Descemet layer is there, even if you get a large hole, you can put it back just at post-operatively. You have to put some air tamponade and be wary of Wurtz-Zavalier syndrome and pressure. So if you put air in the eye, whether it is for DALC or DMEC or DSEC, one has to make sure the pressure doesn't go up and release the pressure measured within an hour of the operation. I think that avoids a double anterior chamber from forming, but I would never convert to a PK.
Wow. Many questions have been well answered by the maestro right now. Wonderful. Fellow ophthalmologists, that's all folks. We learn a lot at this very moment from one of the best eye surgeon in United Kingdom, in Europe, and entire world. Thank you so much, Professor Harminder Dua, for this memorable lecture and useful information you have shared with us this very morning, UK summertime. Thank you so much, Professor.
My pleasure. Thank you very much for having me.
Thank you so much. Thank you so much. And
Bye-bye.
thank you also, bye-bye, Professor. Thank you also, Prima Vision Eye Hospital, Perdami Cabang Sumut. Thank you all the committees, Dr. Dia in Boston and Robbie in Medan, Indonesia. Thank you, my resident of ophthalmology from Universitas Prima Indonesia. And thank you all of my friend, the amazing participant this evening, Indonesia time. And back to Harminder, please send our regard to your family. Thank you
Thank
for
you.
incredible lecture, Professor. Wonderful. Thank
Bye-bye.
you so much, Professor. Bye-bye. Thank you so much, Professor. And everyone, stay healthy, stay wealthy, and always stay happy, everybody. Thank you so much. Wassalamualaikum warahmatullahi wabarakatuh. The next maestro will be Dr. Firna from Indonesia. And greeting from Boston, Massachusetts, and bye-bye.
Life will find a way through night and day, living in true awareness, come what may. We are the man. to those who believe. Step by step we fight with bravery, hearts wide open, never alone. In silence or in battles cry, the answer comes when spirits fly. We strive, we thrive.